Hey guys, how you doing? So welcome to the second part of chapter 2. In this chapter, we're going to finish off the rest of the different organelles inside the cell, and we're also going to have a look at the endosymbiosis theory. Okay, so let's hop into it. We're going to pick up from where we left off last time, and we're up to the next organelle, which is the lysosome. Now, lysosomes are digesters. Yes, they are the organelle that is responsible for digestion or breaking down things within the cell. Okay, they are fluid-filled sacs. In other words, they're a small sac that contains a particular fluid and a large number of digestive enzymes. Yeah, now an enzyme is a protein that is designed to help break down foods and substances into smaller compounds, right? And so a digestive enzyme has the purpose of breaking things down. And so by having a large sac full of these enzymes, it can break down things like the carbohydrates, lipids, proteins, and and nucleic acids, which is pretty much every part of a living thing. Yeah, and so those different compounds form part of living things, and the lysosome is designed to break them down. Okay. Now, they are typically 0.2 to 0.8 micrometers, and remember, a micrometer is 1/millionth of a meter, and they're only active at a particular pH, which is of 4.8. Yeah. Here is the diagram of what the lysosome looks like, and here is what the lysosome looks like under a electron microscope. Okay. So the functions: all excess material that the cell has gets broken down. So if a cell needs to break down its protein for any particular reason, or a part of the cell for a reason, it will send it to the lysosome, and then what happens is the material gets digested and broken down, and then it gets reused by the cells. It's kind of basic components, the basic building blocks like the amino acids or the glucose or things like that, or the fatty acid chains, are then recycled by the cell. Okay. Now, another function that's really important that's done by the lysosome is a particular process called autophagy. Yeah. Autophagy is meaning phagia means to eat, and then auto means self, right? And so autophagy means it's breaking down its own non-functional organelles. Anything that is old or damaged, the lysosomes are actually responsible for breaking them down and getting rid of them. And so you can imagine, in terms of cluttering and decluttering a cell and removing things that are old or dying within the cell, that's what it does, right? So the lysosomes are responsible for those things, okay? The last part is that it also digests bacteria into the cell. And so what happens is, for example, if the cell is a white blood cell, the lysosome is responsible for breaking down the bacteria that the white blood cell catches. And so think of this as like in your blood, you've got the white blood cells as kind of like a police army. And then this part is what actually gets rid of those bacteria and the things that they capture within the blood that doesn't belong to the body. And so that removal and digestion of foreign bodies is also part of the function of the lysosomes. Okay. Now, the autophagia part is quite interesting because during a particular process in development naturally within organisms, there is a process called apoptosis that occurs. Now, apoptosis is programmed cell death. When the cell purposely and intentionally dies, or the organism gives the instruction to get rid of the cell, that's apoptosis. Yeah, and so whenever that occurs, the apoptosis, the lysosomes will release all of the enzymes that it's holding within that sac into the cell. And so what happens is the enzymes start digesting everything within the cell. And so the process is basically a self-killing method. Yeah, and so apoptosis is known as programmed cell death, and when a cell destroys itself. And so if you think about the reasons why a cell would ever need to destroy itself, a lot of it comes down to kind of important parts of development. So I'll give you one example. This diagram on the right here is of an embryo. Now, when the embryo starts to grow, at first it kind of grows up in size, but then as it turns into a fetus, it also then begins to, you'll notice, develop more prominent limbs, like the hands, instead of just being this kind of stubby, this stubby thing that just sort of sticks out of the body, it actually becomes digits. Yeah, and so what happens is you got to remember that stuff doesn't just sort of happen to stretch out on its own, but the cell between the digits, between the fingers, are designed to actually undergo apoptosis and kill themselves, so that you start to have a more prominent digits that come out of the hand, which forms your fingers. And so what happens is the body of the organism programs these things because they're part of development. And so they play a really important role in controlling the depth of zones of cell in the human hand. And so the fingers can then actually become separated until you're left, until you're left with your actual digits and your hands and your feet and things like that. Okay, so that programmed cell death is really important. Now, apoptosis also plays a really important role in managing the body, especially when certain cells in the body have become rampant or out of control. Like, for example, in the case of something like cancer, apoptosis is designed to just prevent the cancer from spreading by just killing the cell that is cancerous. Yeah, and so we'll actually look at that in depth in semester 2, but that's apoptosis. Okay. The next one is peroxisomes. Peroxisomes are similar to lysosomes. Yeah, they're organelles that are also responsible for the breakdown of substances. However, they're not so much used in that recycling as they are in breaking down of toxic substances. Yeah. So, for example, hydrogen peroxide, which is H2O2, right? And so they're designed to break down long fatty acid chains. Yeah. And they're characteristically 0.1 to 1 micrometer in size, a little bit larger or varying in length and size compared to your lysosomes, and they also contain a large number of enzymes. And so they actually break down different things, right? So the long fatty acid chains and toxic substances are broken down by your peroxisomes, and then your carbohydrates, your glucose, your nucleic acids, and your proteins are broken down by the lysosomes. Okay, and here's just a diagram of what they look like. Those little green shiny things are your peroxisomes. Okay. The next one, chloroplasts. Now, chloroplasts are present in plants and algae only, but they are the organelle that is found within the plant and the algae, which is responsible for photosynthesis. So they are like your solar panels, your energy converters within plant and algal cells. Okay, they're relatively large organelles, thought to have once been cells on their own, and they're green because they have a particular pigment inside of them that traps light called chlorophyll. Yeah, so this chlorophyll pigment will absorb and trap light, and it will absorb and trap every spectrum of light except for green, which is why it appears green, right? And so the structure is comprised of these layers called grana. And so a grana is this little green layer or stack that sort of sits there, and then the stroma are the fluid-filled spaces that sit in between, right? And so that's the structure, and that structure allows the chlorophyll to then absorb the sunlight, the chloroplast to absorb sunlight and then convert that sunlight into energy, yeah, as you'll sort of find out later on in the next chapter. Yeah, here is a diagram of what that looks like, and you can sort of see there's your chloroplast, right? And then there are your layers of grana, and then your stroma being the fluid-filled stuff that's sort of in between there. Okay. Now the process is that the chloroplasts use is photosynthesis, and photosynthesis is turning carbon dioxide and water into glucose and oxygen. Yeah, it's what allows plants to build their cells or build molecules from glucose. Yeah, so they take carbon dioxide, 6 molecules of it, plus 6 molecules of H2O, and through the process of sunlight will form 1 molecule of glucose and 6 molecules of H2O, of water. Not water, oxygen. And so you've got carbon dioxide and water forming glucose and oxygen, right? And so that's what the process looks like. And that also allows it to then use as a form of energy or a way of building its own cells, okay? Okay, so the next two, which have sort of been lumped together, even though they are different structures, are the cilia and the flagella. Now, both of these organelles are designed for movement, and they sit on the outside of the cell, okay? And the only difference between them is you can think of cilia as being hair-like structures, and the flagella being a whip-like or a tail-like structure, okay? So if you think of a cell, like for example a bacteria cell or a cell in your body, the cilia are these little hairs that sit on the outside of the cell, and then the flagella is this large tail that it has, yeah? Flagella being plural and flagellum being singular. Okay, and so they're slightly different in the way that they're set, but this is kind of what it looks like. So you can sort of see there. This is a microscope slide of a series of cells, and you can see there that's the cell, and then these little hairs are your cilia. Okay, not flagella; they are cilia. Okay. They are made up of a particular compound called a microtubule, and it forms this kind of elongated structure that you see. And they both function for movement purposes. Okay, the flagella just allows it to move like a tail, and then the cilia kind of have this kind of direction where they beat in a circular motion that allows the cell to move through a liquid or an environment. Okay. Now the hairs also serve other purposes. Yeah. So for example, the hairs can also serve as a way of moving objects along. Like for example, your throat lining will have cilia, which will then beat and move your mucus up and down your throat and things like that. You also have hairs on the inside of your ears, which contains a fluid. And what happens is they respond to sound. The fluid responds to sound, and it responds with the hairs inside the cells of your ears, which is what allows you to hear in the first place. Okay, so very complex processes, but very interesting and fascinating ones. Okay, now these microtubules, this is what it looks like. If you were to cut one of these microtubules up, you would get something like this, and then the cell would have these series of microtubules there, and the cross section sort of shows these circular rods that are sort of stuck together, and they form the cilia or the flagella that the cell has. Yeah, and so that's the cross section that's there, right? And then it's encased in a section of its own plasma membrane there. Okay. Okay. The cytoskeleton. The cytoskeleton is the structural 3D framework of the cell. Yeah. And what I mean by that is it's composed of the same thing as your cilia and flagella, these microtubules, except think of it as inside the cell. It's not like everything just floats freely in a liquid, right? But it actually is strung together in this kind of network within the cell. And that network is called the cytoskeleton, yeah? Cyto meaning cell and skeleton meaning like skeleton, yeah? So cytoskeleton is the structural skeleton of the cell, right? Made up of these microtubules like cables or rods, okay? So it supports and holds the organelles in place. It keeps the cell's main kind of shape so that it just doesn't flatten or change its shape. It allows some sense of mobility by having that particular shape. And then it also allows organelles to move within the cell. Particularly important when chromosomes need to move during cell division. And so when the cell starts to divide, the cytoskeleton plays a really important role to form the spindle fibers, which then allows the process to take place and the chromosomes to move around. Okay, and what it is, it's made up of a series of microtubules, which are your large hollow tubes. And what happens is microtubules are made up of several strands of microfilaments, and then the intermediate filaments, and then the intermediate filaments are made up of microfilaments. Yeah, and so you can think of it like this: microtubule is that, right? And then that is made up of a microfilament, and then an intermediate filament, and each of those intermediate filaments are made up of microfilaments there as well. Yeah, and then each of those are made up of an actin subunit. Okay. Now, in terms of the amount of detail that I need you guys to know about, you need to know what microtubules are, that they're comprised of intermediate filaments and then microfilaments. I don't need you to know specifically the name of tetramer subunits and actin subunits and things like that. Okay. But it's kind of this progression of microtubule, intermediate filament, microfilament. Okay. So it's a very complicated structure, even though these things are really small inside your cell. All right. Okay. So let's put it together. Now, you've basically learnt about all of these different organelles and each of their unique functions that they do, okay? Those functions need to be coordinated and they need to work together for accomplishing a task within the cell. So if you think about the process, right, that they do, each of them sort of all come together and they all sort of work together as a bit of like a kind of mini society or a metropolis within the society to perform the function that it needs to do, right? So this is the summary table that I've just taken directly out of your book. But let me give you an example, okay? Let's say your body, this particular cell needs to produce a type of protein. Like, for example, if it is a protein um, like insulin, for example, right? Let's say it's created by the pancreas. Now, to make insulin, insulin is made up of a protein, right? And so if you go through your different organelles, each organelle will play a different role within the cell to produce that protein, yeah? The first starting point will be the nucleus, yeah? Inside the nucleus has the blueprint, which contains the genetic instruction to make that insulin molecule, that protein molecule, right? But that's not enough. So what happens is the cell then needs to transcribe it and translate it into an RNA strand, and that RNA strand then needs to leave the nucleus and goes to the ribosome. Now the ribosome is the protein factory. So the ribosome will use that RNA strand, and then it will collect the necessary amino acids into the ribosome, and it will assemble the amino acids into a protein or a polypeptide chain. And then what happens is the polypeptide chain then gets transferred to the ER. Now the ER would be either rough or smooth. In this case, because it's a protein, it would be the rough ER. So the rough ER would then fold the polypeptide chain into the protein shape that it needs to form, and then it will then send it, let's say it needs to send it outside of the cell. It will send it to the Golgi complex. The Golgi complex will package it, and then it will send it out of the cell. Now, it will send it out of the cell through your little vesicle, and then it will leave the cell through the plasma membrane, okay? Now, all of that process requires energy because the cell needs energy to do that. And in order to do that, it will use energy that it receives from the mitochondria, and it will use ATP that mitochondria has produced to do that, yeah? And so you can sort of see there, very simple process of, very straightforward process of producing proteins that requires, all 5 of these organelles to function, okay? You can say the same thing about when the cell needs to divide. It needs to use the nucleus, the cytoskeleton, and all these other cells. If the cell needs to, control or do some sort of digestion, it will need to use the lysosomes or the smooth ER and things like that. So each organelle works together with other organelles to perform the unique function within the cell, okay? All right, we're going to end with the endosymbiosis theory. Now, the endosymbiosis theory was developed by a particular scientist named Lynn Margulis from 1938 to 2011. And she proposed this theory that was sort of began in 1967, that some organelles were actually once living organisms, and they were eaten by the eukaryotic cell. So in other words, organelles such as chloroplasts and mitochondria are actually quite large compared to some of the other organelles, and they almost have mini structures of their own. So they're organelles that have almost like their own organelles. And so Lynn kind of proposed this idea of the endosymbiosis theory, yeah, which means that they were once free living bacteria and lived on there independently, but then they were absorbed through cytosis, which would be endocytosis, into a eukaryotic cell. But then instead of digesting and eating them, they actually kept them and incorporated them to become part of the cell, and therefore developing a complex eukaryotic cell, right? And so the word endo meaning inside, symbiosis, sim meaning together, biosis meaning life. So life, so basically one life within another life, and so that's what the word endosymbiosis means, okay? And so it's a very interesting theory proposing that, and you can sort of see the complexities of a eukaryotic cell compared to a prokaryotic cell, because the eukaryotic cell may have absorbed these chloroplasts and these mitochondria into themselves and incorporated them as part of their being, right? So that's the endosymbiosis theory. Very, very interesting. So the benefits for both cells, the eukaryotic cell will actually get free energy from the chloroplast bacteria, and the chloroplast bacteria has a more efficient method of producing that energy because it has now a cell that it lives inside that will protect it, give it the nutrients and the substances it needs to produce that energy, and then there's this kind of mutualistic benefit to both the chloroplast and the cell that holds it. right? Possible evidence. So chloroplasts and mitochondria both contain circular DNA. So the chloroplast and the mitochondria actually has its own set of DNA, unlike the ribosome or the Golgi complex, which doesn't have its own DNA. But the mitochondria and the chloroplast do, and so and it's circular, which is very similar to the DNA of bacteria. And so there was the evidence is that this may have been a bacteria at one point and then absorbed in. Okay. They also reproduce by a form of binary fission, which is also similar to bacteria, where they split apart and multiply when the cell divides. And they're also very similar sizes to bacteria. And so those are some of the evidence. You can also, other evidence include elsewhere, for example, in a lot of plant cells, you have bacteria that are nitrogen fixing already living inside the cells of these plants. And so that's additional evidence for endosymbiosis theory that things can be absorbed in and live inside something else. Another really good example is single-celled algae living inside of the cells of corals. And that's the reason why corals, even though corals are animals, can photosynthesize and have the property of plants, because it's not the coral cells themselves that do it, it's the algae that live inside of their flesh, right? And so these single-celled algae live inside the corals and will do the photosynthesizing for the coral, and then in turn the coral will give them the nutrients and things like that. Very fascinating theory, a lot of evidence for it, and very compelling. Okay, all right. So we're going to finish it there, and then next lesson, next video, we're going to look at the macromolecules in biology. Okay, all right. I'll catch you guys later.